So our video today is all about DNA replication. What is DNA replication? DNA replication is the process by which a cell makes a copy of its DNA. So, what are some basic facts about DNA replication? First, it happens during the synthesis phase of the cell cycle. So if you remember the cell cycle, we have G1, we have S, we have G2, and we have mitosis. And S stands for synthesis, and what happens during that synthesis phase is DNA replication. And the nice thing about DNA is that DNA can serve as a template. That means that we can build the new strand on the original strand. We can use it to tell us what should go where. There are enzymes and other proteins that do the actual work of replication. And those enzymes are going to have to do several different things. They're going to have to unzip the double helix, and they're going to have to put into place free-floating nucleotides and form hydrogen bonds with the template strands. So here we have a little diagram of that DNA being unzipped. And you can see we split apart the DNA strand and we're putting in our free-floating nucleotides. Those are going to form hydrogen bonds, and then enzymes are going to go through and they're going to seal the backbone, which means they're going to connect those phosphate groups with those deoxyribose sugars and make a solid backbone. So let's kind of go through this process. The first thing that's going to happen is an enzyme called DNA helicase is going to unwind and unzip the DNA. And then another enzyme is going to go through called DNA polymerase, and it's going to bond the nucleotides together with the strand of DNA. And you can see that happening right here. So we have our enzyme and we have our free-floating nucleotides. They come in, they bond, and then the polymerase seals the backbone and makes a solid strand. And here is what our two replicated strands of DNA look like. These are our copies. And you can notice that each one has an original backbone and a new backbone. That's because DNA replication is a process we call semi-conservative. That means we hold on to one of the original strands and we also build a new strand on top of it. And we're going to be using those base pair rules to formulate that new strand. That's why we call this a template, is because it has the sequence of DNA, and then the new nucleotides can come in and bond following the base pair rules, A to T, C to G. DNA replication doesn't start at one end of the strand of DNA and run to the other. That would take forever. So instead of doing that, we have what we call replication windows. That means all along this strand of DNA, we are replicating in different spots, and we're building out from that starting location out until we meet up and we have two fully replicated strands. Okay, that is all we're going to cover with DNA replication. Very quick, very simple. We're just opening the DNA strand, building our replicated strand right there on top of it, and we end up with two copies. And that's what we're going to need to pull forward as we move forward with this unit.